Hello, my dear students. I hope you all have been doing well. Today, I am going to deliver you lecture number 6, and this lecture is the part of the topic of drug metabolism and drug design. Actually, this topic was based on two lectures, that is lecture number 5 and lecture number 6. And I I break this topic into two lectures because it was a vast uh, topic in that we have to understand how the drug metabolism and the drug biotransformation knowledge would be helpful for designing of a drug. So in the part 1, we have already understand what is the drug metabolism. We also say called drug metabolism as drug biotransformation. So in lecture number 5, I have already explained you everything about drug biotransformation or metabolism. That is what is the drug metabolism or drug biotransformation. What are the phases of drug metabolism? What are the reactions of drug metabolism? What are the consequences of drug metabolism, and what are the root of drug metabolism. Now in this lecture we would be studying how the knowledge of the drug metabolism that we have already discussed in lecture number 5 would be helpful for us for the designing of a drug. So uh, as I already have discussed with you about the drug metabolism, but I will conclude you here again for your ease that drug metabolism is a change, any change in the drug. What is the change? Change is the means change is for example, if a drug is active, and it may be possible that after biotransformation of drug metabolism it become inactive, or an inactive drug may become active, or maybe it may be possible the drug remain unaltered. So was the drug metabolism, and drug metabolized by different reactions. They may be oxidation, sulfation, methylation, acetylation, and glucuronidation, and all these reaction uh, takes place either in the liver microsome or in cytosol. So the all the reaction that takes place in the liver, that are liver microsome, that are phase 1 reaction, and in cytosol there will be phase 2 reaction that will be takes place. And in phase 1 reaction, the drug will be metabolized in such a way that it will be add up a polar functional group in that. So so the drug will be more polar and more polar. And the phase 1 reaction actually takes place in the drugs, so it make drug more means polar form, so it will be easy to metabolize in this phase 2 reaction. So phase 1 reaction need is what is the need of a drug to metabolize in phase 1 reaction. The phase 1 reaction metabolism in needed are needed for phase 2 reaction metabolism. Phase 2 metabolic reaction. So means for the phase 2 reaction, drug must be previously present in sufficient polar form. So this is the reason of phase 1 reaction. So and in when drug metabolized in phase 1 reaction, and then it reaches to the phase 2 reaction. So what is the purpose of phase 2 reaction? In phase 2 reaction, drug become more polar or sufficiently polar, so it can be excrete out from the body. Because I have already told you that the drugs that are non-polar in nature, and the drug that are lipophilic in nature cannot be excrete from the body. For the drug to excrete from the body, it should be more polar in nature, so it make a bond, means it can be soluble with water. Polarity is what? Polarity is the solubility. If a drug is polar, so it can be easily soluble in water. So we phase 2 reaction make drug more water soluble by adding the polar functional group in that, or by adding doing the glucuronidation. So what will happen, the drug get more polar and more soluble in water, and then they make the phase 2 reaction, make the drug more lip hydrophilic. So it will be hydrophilic, so or hydrophilic means water loving, so it will be hydrophilic, so it cannot be reabsorbed again into the lining of the kidney tubules, so it cannot be back into your general circulation. So it, if the drug will be more hydrophilic, so it will be water loving. It will be water loving, so it will be soluble in water, and it is radially excrete out from your body. So the excretion is one of the example of the one of the need of drug metabolism. Why drug metabolize for excrete from the body, drug metabolize to become more biologically active, drug metabolize to become more biologically inactive, and drug metabolized to remain unaltered. So these are the purpose of drug metabolism, and excretion is the one purpose. Because of that we need a drug in its metabolic form. Why we want to we want a drug to be excreted from the body, so we want that this drug metabolize. And why, what is the way the drug metabolize we need? We want that drug metabolize in such a way for excretion, so it can add up a functional group in that. So if this functional group should be polar and make the drug more polar, and drug soluble in water and excrete out from our body. So now in this, I give you again a conclusion of the drug metabolism. So it will be helpful for you to understand this lecture. Now in this lecture, we understand that how this knowledge of drug metabolism that I have already right now explained you here would be helpful for us to design our drug. Now, so this knowledge of drug metabolism, what how we can use the knowledge of the drug metabolism for designing of a drug. For example, if we are taking here an example of a drug that is built upon a phenol moiety. So just imagine that someone is having a, a disease, a chronic disease, that is a cancer, and you have to administer a drug to this cancer patient, and you are having an anti-cancer drug, and this anti-cancer drug is built upon a phenol moiety. So what will happen when you administer this drug to the cancer patient by oral route? So you have knowledge of drug metabolism from a previous lecture. So what will happen, where the drug will go from oral route to the intestine, and from intestine it will go to the portal circulation, and from portal circulation it will go to the your liver before reaches to the target site. So what will happen in liver if the drug will reaches there? In liver, I have already explained you in my previous lecture. In liver, what will happen, the drug get metabolized. Where the drug get metabolized, in microsome or in liver microsome or liver cytosol by the action of enzymes cytochrome P450, by different reactions, oxidation, methylation, acetylation, and glucuronidation. So what happened with this phenolic drug, phenolic anti-cancer drug? If you administer this phenolic anti-cancer drug with a cancer to a cancer patient, this drug will go from your oral route to the intestine, from intestine to the systemic to the portal circulation, and from the portal circulation to the liver microsome. So the what will happen in liver microsome, the phase 1 reaction takes place, means the functional group will be added to the phenol moiety. And then what happened, and then drug will be available to metabolize for phase 2 reaction, and it will go to the liver cytosol. So what will happen in liver cytosol, the phase 2 reaction takes place with the phenolic anti-cancer drug. And what are the specific reaction that will takes place with phenolic drugs? uh, what is the particular phase 2 reaction that will happen with phenolic drugs in liver? It is sulfation, and I have already discussed with you in my previous lecture that in phase 2 reaction, what will happen, there is a reaction of sulfation, means addition of sulfate group to a phenolic drug. So what will happen with this phenol drug? This phenol drug will reach to the liver cytosol after phase 1 reaction, and in where there in liver cytosol phase 2 reaction takes place, and in phase 2 reaction what will happen, there will be a sulfate group that will add up in the phenol group. So what will happen then? After the addition of this sulfate group, the drug become more polar and then radially excrete out from your body. But you don't want it, because you want to administer drug with to a anti-cancer patient, because this is a anti-cancer drug. So if you will administer this drug to an anti-cancer patient, and it will radially excrete out from your the body of the patient, so what would happen? You will not will be having a proper mechanism of action or a longer duration of action of this drug, because cancer is a chronic disease and you have to administer such type of a drug that can remain in the body of the cancer patient at the site of the at the target site for longer period of time, so uh, they can deliver its sufficient mechanism of action for a longer period of time. So what you will do with this anti-cancer drug, because you are not achieving your purpose is that you administer a drug to anti-cancer to a cancer patient, and that drug remain in the body of a patient of a cancer patient for a longer period of time, and then uh, produce its mechanism of action at the target site for a longer period of time, so the patient can be treated sufficiently. But it is not happening. So what, because the drug is getting metabolized. Why drug is getting metabolized? Because a drug is built upon phenol moiety, that is adding up a sulfate group during a phase 2 reaction and getting out from your body from the body of the patient so quickly. So what you will do in this situation, being a pharmacist or being a drug designer, you will think about that you build a drug or you design a drug, a anti-cancer drug in such a way so it is not having phenol moiety. It is not built upon on phenol group. It is built upon any other structure or any other moiety that if reaches to the liver, so it cannot metabolize by liver's phase 2 reaction, that is sulfation. So it cannot be it would not be excrete out from the body radially, and it can give you a longer duration of action. So this is how the knowledge of the metabolism help you to design a drug. So this is the one example. So now if you are a drug designer or you are a pharmacist and you are designing of a drug, and how all these changes in a drug can you are doing the changes in a drug by changes the structure of a drug, means you are making a short duration of action, a drug that is having short duration of action. You are making this short duration of action of a drug to longer duration of action. How you are doing this? You are doing the activity of a drug by converting the structure of a drug. Okay, so what the consequence you are getting for it. You are by doing structural changes, you are making the drug more stable. Means if you are changing the phenol moiety from the drug, and you are designing a drug on another moiety, so what will happen, the drug will not get sulfate, will not do sulfonation in the liver, and it will be more stable. If it is not doing any reaction, that is sulfation or any other reaction, means it is more stable. So by the drug designing, what you are doing, uh, drug designing is the structural changes of the drug, and by this drug designing you are making drug more stable. And what are the analogs? Analogs are the, means if you are having a particular drug, previously parent compound, but you are having this similar drug that is having similar mechanism of action but having different structure, so it will be the analog of the previous drug. So what you are doing, you are making an analog of the previous drug by doing structural changes in the drug, and this analog is more stable, so it is cannot be metabolized by liver phase 2 enzyme and cannot excrete from your from the body of the patient, and giving the longer duration of action. So this was the one example that by doing structural changes in the drug or by drug designing, uh, you can make drug more stable. But in many cases you can also make drug unstable. So you can make a drug by drug designing that can be easily liable for the drug metabolism. So uh, what cases in this, you can make the drug more unstable? When you needed a drug, uh, 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 eh, when you needed a drug that you want that this drug excrete from the body radially, so what you will do, you will design a drug in such a way so it can be more stable, so it easily metabolized in your liver. And if when it will be easily metabolized in the liver, so it will be as easily excrete out from the body. So one important thing is that while doing structural modification or while doing drug designing, you should have to keep in your mind that you should have to design a drug in such a way that it your modification does not change the biological activity of a drug. For example, I have given you an example of anti-cancer drug. If you are doing structural changes in an anti-cancer drug for making it more stable, but you have to do these structural changes in a such a way that you just make drug more stable. Means if you are having an anti-cancer drug, after your structural changes the drug remain anti-cancer. Means your modification are not in such a way that your modification change an anti-cancer drug to an for example antipsychotic drug. So you have to the structural changes that someone is making in a drug during drug designing, or some if it should be the structural changes should be in such a way that it should not affect the biological activity of a drug. And how you know that the biological activity of a drug could not get be changed after your modification. You will find out it by doing some experiment that will tell you about the biological activity of these drugs. For example, there are various experiments. For example, one experiment is anti-migration assay. If your drug is anti-cancer, it is having means it is having and it is anti-migration agent also, means anti-metastatic agent also. So you can perform anti-migration assay, you can perform anti-cancer assay. So you know the cancer is a disease that is in that the cancer cell having the ability of migration, means for reaching from one side to another other side, and cancer cell having the ability of proliferation, and cancer cell are having the ability of not die, means they not die, they remain survive. So if your drug you are making any structural changes in the drug, so you should have to keep in your mind that uh, your modification does not change the biological activity of the drug, and you how you can identify that after your modification your drug have not lost its biological activity. So for this purpose you will perform different experiment. In the case of anti-cancer drug, you can perform an cytotoxicity assay, you can perform the anti-proliferation assay, you can perform anti-migration assay, and like that there are various assay that can tell you about the anti-cancer activity of this drug. Now we will see that what approaches we can use for the designing of a drug based on this knowledge of drug metabolism. So first approach is this: we can increase the metabolic activity stability of a drug, enhance the duration of our action. So first, what is the first approach? In first approach, we design a drug in such a way that we will be having a drug in its stable form, so it cannot be acted upon the liver metabolic enzyme or the liver metabolic reaction. So if it will not be metabolized in liver, or it will not do reaction, it will not do, or it will not participate in phase 1 and phase 2 reaction of liver. So what will happen, it will be having longer duration of action. So what we can do, or we can how we can design a drug for this purpose, or for this for the or by using this approach. In this approach what will we do, we will replace a drug more reactive group with the less reactive group. For example, if we are having a drug, for example an anti-cancer drug, and it is having a ester functional group, ester functional group, and then this ester functional group is more reactive, and when it reaches to the liver, and it will do esterification in liver, phase 2 reaction, and then excreting the drug in the body. So what we will do, we can remove the ester functional group from an amide group. So here we are having an standard rules, means we can replace ester functional group from every drug by amide. We cannot means remove ester group by any other group. But if you are having a drug and it is having an ester functional group, and drug is more reactive with liver phase 2 reactions or liver phase 2 metabolism, so what we will do, we can replace this liver this ester functional group with the amide group. And other approach is, if a drug that we are having, and it this drug is having a nitrogen atom in its ring skeleton, and this nitrogen atom is having alkyl group. So and this drug if reaching to the liver, in liver it can do the it can remove the alkyl group from it and become less reactive. So what we can do, we can prevent this end dealkylation by removing alkyl group at the end position of the drug to the methyl group. And we can also prevent the oxidation of all those drugs that are having aromatic ring in its structure. So by introducing the strong electron acceptor such as chloride, quaternary amines, carboxylic acid, sulfonate, and sulfonamide. So this was the first approach. In first approach, what we do, we make stable, means we design our drug. In first approach, how we can design a drug uh, that is more stable and then less likely to be acted by liver metabolic enzyme or liver metabolic reactions, or give us more longer duration of action. So how we design such type of a drug, we make changes in the drug skeleton or in the drug structure in such a way that we replace a reactive functional group from less reactive one, or we can prevent the oxidation of some group in the drug by removing the by introducing the strong electron acceptor group, such as I told you that chloride, amines, carboxylic acid, sulfonate, and sulfonamide. So this was the first approach, and here we are again taking the example of first approach. What is the example? The example of the conversion of tall butamide. What we will do here, we replace the aryl methyl substituent of the anti-diabetic tolbutamide by chlorine. So what will happen, that it will aid the anti-diabetic chloro chlorpromide, a compound with a considerably longer half-life. So what do with this example here, we were having a anti-diabetic drug that was the tolbutamide, and this tolbutamide drug would be having here this tolbutamide drug were be having aryl methyl substituent. This is aryl methyl substituent. This is methyl and this is aryl group. So this drug was having aryl methyl substituent. So what we did with this drug, and this drug was having shorter half life. So how we designed this drug? What we did, we replaced this aryl methyl substituent and we add the chloride group here. You can see here, means this drug was having a methyl group at this ring. So what we do, at this aryl structure it is having methyl group, we remove the this methyl group and this we add the chloride group. So what happened, this drug become chlorpromide, although it is having the same anti-diabetic activity. So but by the changing of the functional group, what we change, we change the functional we change the half life, we change the duration of action of this drug. So this is the one example, and here we can taking the second example. Sometime sometime the change of the functional group can change the biological activity or pharmacological activity of a drug. For example, if we are having a drug that is a procaine and it is having a ester group, but when we remove this ester group from this procaine and we introduce an amide group, so what will happen, we will be having a drug that is a procanamide, and it is anti-arrhythmic drug. So what will happen, by what we can do during the drug designing, we can change a functional group, so we can get any other new drug. For in as in the case of procaine, that is having a ester group. When if we remove this ester group from this drug and we add an amide group, you can see here C double COOCH2. So what we introduce between this group? We introduce amide group NH. You can see here we introduce NH. So what it become? It become procanamide from the procaine. So procaine was having what activity? Anesthetic activity. But when we add amide group in procaine, it become procanamide, and now it become anti-arrhythmic drug. So by using this approach, we can also by using first approach, we can also change it may be possible by using the second approach, the biological activity or the pharmacological activity of drug become changed. Now we are going to the second approach. What is the second approach? We can make drug, we can we can increase the ease of the metabolism of a drug by using the second approach. How we can do this? By incorporating a metabolically liable group such as an ester in the structure of a drug. Means by using the second approach, we can make all those drugs that are not easily metabolized. We can make all those drugs in we can design or means convert all those drugs um, in a metabolically unstable form. Means if we are adding ester group in a drug, so it will be easily metabolized in the liver. So these are biologically active compounds that are rapidly metabolized by a predictable route to pharmacologically non-toxic compound. So here is a disadvantage of this approach, that that is the half life is so short that the possibility of the patient receiving fatal overdose is considerably reduced. Means by using the second approach, what we can do, we can make a drug more metabolically active. So how we can do this? We can add a functional group, that is a ester group in that. So what would happen? The drug get uh, easily metabolized in the liver and easily excrete from the body, or having shorter half life. But what will be the advantage of this approach? The advantage will be the drug will be having a shorter half life. So if a patient receive fatal overdose, so we can reduce this fatal uh, overdose of the patient. So we are here having the example of the second approach. The example of the second approach are the neuromuscular blocking agent, that is succinylcholine. This drug is almost fully metabolized by hydrolysis in about 10 minutes, which considerably reduce the chances of a fatal overdose. But the drug can still be fatal, as some people do not have the esterase necessary for hydrolysis. Now we are having the third approach. What is the third approach? In third approach, we can change the drug, the nature of a drug in such a way it change its biological activity. Means we can change the by the drug designing, we can change the biological activity of a drug, but it is this approach having a drawback. Uh, and the drawback is that when we introduce a liable group into the structure of a drug, so what will happen? the biological activity get changed, and we sometime we does not want that we have a drug. For example, if we wish to make a drug, anti-cancer drug, in such a way that want that this drug remain in the body for a longer period of time. But what will happening, we are making a change in this drug, we are introducing a liable group in this drug for the purpose that this drug cannot be metabolized by liver enzyme and remain in the body for longer period of time. But what our changes are doing, what our functional group that we added is doing, the functional group is changing the biological activity of the drug. For example, it is making the anti-cancer drug to the anti-arithmetic drug. This is the drawback of this means approach, because if we want a drug that remain an anti-cancer drug, and uh, our addition of a functional group is changing its biological activity. So we don't want to change the biological activity, but by using this approach, what happened, if we wish to change the nature of a drug, the biological activity must be changed. So this is the drawback of the third approach. And what is the fourth approach? In the fourth approach, we change the metabolic pathway of a drug. What you understanding, uh, what we are doing, we are changing the metabolic pathway of a drug, means we are making a drug in we are designing a drug in such a way that it will not be go through the particular route of drug metabolism, means it will avoid to go through the particular route of metabolism, so it will avoid the drug metabolism. So this approach used to develop analog that do not have the undesirable side effect of the lead compound. For example, the local anesthetic lignocaine is also used as an arithmetic. In this respect, its undesirable convulsion and emetic side effects are caused by its metabolism in the liver by dealkylation to the amino N-alkylation to the mono and ethyl derivative. The removal of the N-ethyl substituent and their replacement by N-methyl group give the antiarrhythmic tocanin, tocanin D. So here is the example. Tocanin neat cannot beta cannot be metabolized by the same pathway as lignocaine and does not exhibit anticonvulsant and antiemetic side effect. So, dear student, thank you very much for your kind attention, and I hope you have understand how the knowledge of drug metabolism is useful to design a drug of your desirable biological activity or desirable mode of action. Allah Hafiz.